You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. And I have two interesting researchers I'm talking today. I hope I get their names pronounced properly. Xiaoyang Wu and Ming, both at the University of Chicago. We're going to be talking about CRISPR, gene editing. And they're going to be talking about strange things like cocaine-proof mice and cracking the puzzle of addiction. So thank you guys for coming. I appreciate it.
Thank you for the opportunity.
Yeah, thank you.
Yeah. So tell me, I've heard in the news a lot about CRISPR-Cas9. What is it? Can you just define it for listeners so that they understand?
Xiaoyang is the best to explain this.
Okay. CRISPR is a kind of, we can see the revolutionary new tool for the genome editing. So that provide a wonderful tool to precisely edit the cell's genome. And we can modify the genome by, for example, correcting mutations. And also it provides us tool to insert like a coding sequence of therapeutic genes into a safe locus precisely into the engineered cells.
So as far as I understand, CRISPR-Cas9, it comes from bacteria. It's a mechanism that bacteria use to edit their own genes, right?
Mm-hmm. Yes. It's from bacteria cells, yes.
Okay. So what does it do if you introduce CRISPR-Cas9 into an organism like a mouse? What does it do? Does it go to the coding sequence of the genome and cut out certain parts of the sequence and replace them with something else, or how does it work?
Yeah. So the system depends on two major components. One is called the Cas9 protein, and the second is a guide RNA. So the guide RNA will provide the sequence specificity. So they only recognize a special sequence. It depends on the design. So you can select literally almost any sequence in the mammalian genome, like in the mouse cells. We can select almost anything as long as it has a specific, we call the PAM sequence at the end. And the gRNA will be able to guide the Cas9 to target to that locus. And the unmodified Cas9 protein will be able to cleave the DNA sequence, create what we call double-strand break. And if that happens, the double-strand break may induce we call the non-homologous end joining, and that can cause most of the time mutation in that specific locus. So in this case, it can be used. The tools can be developed based on this concept to repair potential mutation. For example, in the inherited genetic disease, we can knock out a specific, like say, mutated exon. So we'll still make the protein to be expressed relatively at the normal level. So it provides a tool to make the correction. And the second, the double-strand break, when it happens in the genome, that can greatly increase the chance for homologous recombination if the nuclear has enough template, has enough template inside the nuclear. So the damaged the chromosome will be able to use those template to repair it. And in this case, we can insert a potential interesting gene, like a therapeutic gene, into that double-strand break point. So that can provide a tool that we can guide any insertion, like exogenous gene, into a safe locus, into the cells. So unlike a virus-based therapy, that insertion is random and it may cause, mutation to the genome. We call the genome toxicity. And the CRISPR-based modification will be sequence-specific and can be safe.
How do you know that viruses randomly insert their genetic information into a host? They must be doing some targeting.
Yeah, there will be some preference, but it's nearly impossible to control where the virus will insert.
Okay.
Yeah, that's
So this is
the
possible.
very significant barrier for the viral-based therapy. Yeah.
Well, thank God. Yeah. Otherwise, viruses would really cause us big problems, I guess, if they could select it.
Yeah, right now, a lot of people use relatively safe virus. I mean, right now in the field, most people use the adeno-associated virus, so the insertion is minimum, and it can be safe. Yeah, but still, I mean, the expression using the AAV-based platform is more transient expression without insertion into the genome. And if it insert into the genome, we cannot really control the insertion.
Okay, so it sounds like CRISPR, you'll cut out a coding sequence, and then will you let the organism itself repair the damage, or will you also insert a new sequence that you want to be expressed, or can you do both? It just depends.
Yes, not necessarily cutting the coding sequence. It can target intron and it can target intergenic region in the chromosome. It depends on the purpose of the modification.
Yeah, can I make a comment there? So Xiaoyang is absolutely right. So the CRISPR allow you to do at least two different things. One is to cut coding sequences to repair the mutations, or in our case, it's to deliver the gene to what's called a safe location where there's no gene expression, so the gene can be targeted there and expressed without affecting the endogenous gene. Sorry, just a little bit clarification.
No, that's fine. Okay. So what are some of the examples of using CRISPR, and what kind of effects have they had?
Can you repeat your question?
Yeah, and are there what specific examples are out there of where scientists have used CRISPR? What kind of sequence have they removed, and how has it affected the creature?
Yeah, for example, there are multiple companies right now doing the CRISPR-based gene therapy clinical trials. As I remember, I think Editas is trying to use the CRISPR to correct mutations in its inherited eye disease. So I think the basic idea is using the CRISPR to target the retinal cells so it can remove mutated axon and can allow the gene to still be expressed. Without that gene, the patient will eventually go to blindness. Yeah. I think the second, they also try to use CRISPR to correct the sickle anemia. So that's a mutation in the red blood cells. Yeah. There are many potential application for using the CRISPR technology. It's really a wonderful tool. Yeah.
So how are you guys working with it specifically? What's your research about?
I think Ming can probably talk more. So our studies try to focus on the addiction and using skin as a vehicle. Yeah.
Okay, I can comment a little bit, but Xiaoyang certainly started this whole thing, and he has used the CRISPR method in mouse models to correct a mouse model of diabetes, and he can comment on that. But the collaboration between Xiaoyang and I, our two labs, targeted drug abuse in general. But the first example is to potentially develop the first approach to treat cocaine abuse. When I say first, I mean there are no FDA-approved medications for treating cocaine abuse, and unfortunately, cocaine addicts, they are turned away from the treatment centers and hospitals and so on and so forth. And the basic of it is that the essence of this approach is that the development of this approach is aided by three advancements. The first one is there is a naturally occurring cocaine-degrading enzyme we call the BChE, and there are smart scientists who recognize this because the enzyme can degrade cocaine. They have been doing what's called computer-directed mutagenesis, and enabling over 15 years of research, enabling the resulting protein to have a 404,000-fold increased cocaine-degrading enzyme activity, and at the same time reduce the normal enzymatic activity for its normal substrate called acetylcholine. So that's one development. A second development is, we covered it already, CRISPR. So CRISPR technology allows us to target this modified BChE gene to a safe locus where the introduced gene can be expressed nicely without affecting, hopefully, the expression of all endogenous genes. And the third development, perhaps the most important development, is occurred through Xiaoyang and Xiaoyang's work. That is, he was able to isolate skin stem cells using using CRISPR to modify the genome of the skin stem cells, and then he can graft these genetically modified cells into what's called a normal host. Normal host meaning immunocompetent host. So that's very significant because you don't have to be immune compromised in order to receive these cells in a somewhat permanent way. So the three technological advancements allowed to us to test this cocaine treatment strategy. And so what we did was we got this modified cocaine-degrading enzyme, and Xiaoyang targeted it and grafted it to normal mice. And we evaluated using the resulting grafted mice all core features of cocaine abuse. The first one is getting addicted. So you can use normal mice, graft these genetically modified cells, and you can never induce cocaine abuse. So in a way, these grafted mice are protected from developing cocaine abuse. And the second key aspect, which is probably the most relevant to clinical treatment, that is, we can induce cocaine addiction in mice, and then we can graft these cells, and then they can never relapse into cocaine use. That's highly significant clinically, potentially. And the third aspect is, go ahead, question?
Oh, go ahead with the third aspect, and I'll ask you a question.
The third aspect is cocaine overdose, acute cocaine overdose. So there are cocaine naive people or repeated cocaine addicts who repeated use cocaine, and sometimes they get accidental overdose. Many people die. Many people fortunately recover, and there is just no handle on treating acute cocaine overdose. So we found that upon grafting these genetically modified cells, mice are fully protected from cocaine overdose. We can give them twice the lethal dose of cocaine. These grafted mice behave very normally, whereas what we call the control mice, who don't have the proper genetically grafted cells, they die within minutes. So these are the three core features of cocaine abuse. And by grafting of these genetically modified skin cells, these mice are fully protected from these three aspects of cocaine abuse.
Huh. How do you introduce the graft? Is it just, can you just do an injection of CRISPR and it goes to work and does all the grafting for you, or how would you do this?
Oh, no. Xiaoyang can explain this. No.
So I basically I'm a skin biologist. So the reason we choose skin as a kind of receptor to take the engineered cells is, skin is the outer surface organ, so it's very easy to do the grafting and to monitor the tissue. And probably you don't know is so the skin grafting has been done clinically a lot because the bone wound patient for treatment of bone wound, the patient will require transplantation of autologous skin, either from the patient itself, or we can use the cells, the skin stem cells isolated from the patient, and culture those cells, expand the cells in vitro, and generate a kind of artificial or we call test tube skin, and for transplantation. So that's a simple surgical procedure to transplant the engineered skin epidermal tissue onto the patient. So that's why we think that create a really unique window, so we can carry out CRISPR-based genome editing in cultured skin stem cells, and then generate engineered skin epidermal tissue for transplantation to the same patient for the treatment.
Well, for something that acts systemically, how would you influence it? If something acts locally, let's say, I don't know, I have a condition that causes me to have a skin lesion that is cancerous, and you could, let's say, cut out that piece of skin and put in a genetically modified piece of skin. Okay, but what if something acts, again, systemically? How would you affect the whole organism and change its DNA? so that it would be resistant, let's say, to cocaine, or it would overcome some kind of other illness?
Mm-hmm. So skin is although it's a peripheral organ, but it's highly vascularized. So the blood supply and blood circulation is very robust in the skin tissue. So we propose is the protein secreted out from the skin epidermal cells will be able to pass the barrier and enter circulation. So that's why it can cause systematic effect. So for instance, in our paper, we showed the data is we can detect the significant serum level of the enzyme we inserted into the skin cells. So suggest the secreted enzyme will not only stay locally, but enter into the circulation to achieve the effect. Yeah.
But how long does it take for it to start acting systemically? And it would have to, I mean, would it have to influence every tissue in the body, or would it only influence certain tissues? And I mean, that's a dramatic change to the organism to do such a thing.
Yeah, I understand your question. It's for this type of therapy, potential therapy to function, it has to enter into the circulation. You know, that's where the cocaine may enter into the brain after administration. So the enzyme expressed in the skin cells has to be released into the circulation to be able to function. So that's if it stay locally, it will not function. But I mean, express a kind of exogenous or mutated enzyme in the blood may cause potential side effect. But based on the biochemical analysis, we know the enzyme is specially engineered, so it has no its own original substrate. It cannot act on its own original substrate. And we also, based on literature and also our own study, show to the level we tested, the enzyme level we used, it will not cause any noticeable effect on the mouse behavior or any other significant side effect to the animal.
But literally, how did you create cocaine-proof mice? Did you graft a new piece of skin onto the mouse, and the enzyme systemically altered all the tissue of the mouse? You know, the cells that make up the blood vessels, the cells that make up the organs, the cells that make up the skin. Were they all changed?
No, only the
time,
cell, only the cells, only the engineered skin stem cells has the engineered genome with the BChE gene and with that enzyme expression. But that enzyme, the protein product, will enter into the circulation. It will not cause any change to the genetic material in any other cells. It's only the protein product will be in the blood. And in this case, the protein will. Yeah, I mean, do you want to comment on that?
Yeah, can I? Yes. So the clever part of this is, as Xiaoyang described, the gene. So we're using. So this, first of all, this skin cell based therapy is a gene therapy. The only place where there's new genes introduced is this piece of skin. So what Xiaoyang did was to cut a small piece of skin from a donor mouse, and then use CRISPR to target this gene in and amplify the cells a little bit, and put these genetically modified cells as a piece of skin back into the donor mouse. So only a skin patch contains the genetically modified gene. But what's clever, as I said, was that, and also as Xiaoyang said, these genetically modified skin cells is able to express or produce these proteins, and the proteins get transported to the blood circulation. And there, only the protein, not the gene itself. The gene is permanently integrated only in the skin patches.
Okay.
That is the protein that's circulating in the blood. Once you're in the blood, the protein will go to everywhere where blood reaches. And for cocaine abuse, the cocaine reaches a central reward sort of a center and started the behavior, let's say addiction behavior there. So, but before the cocaine can reach there, or when the cocaine reaches the reward system, the enzyme that's in the circulating blood actively degrades the cocaine very quickly, and we know this. We have biochemical data. So it never accumulated enough to produce either a lethality, that is overdose, or have enough sustained effects to induce addiction or relapse. So the protein reaches everywhere.
I understand. Yeah, I understand now. Okay. Well, another question. So what about the surrounding skin? let's say you affected a patch of 1,000 skin cells. Those skin cells, as they divide and make new skin cells, they would carry, I would think, the genetic modification, right?
You mean the surrounding skin?
Well, two.
So the question is, sorry,
cells that you altered and the surrounding skin. First of all, will the skin cells that you altered, as they divide and make new cells, will they carry the modification? And then will it propagate to the surrounding skin cells or not?
It will not. It may self-sustain to maintain the skin graft, but I mean the chance the modified genome to to enter the adjacent normal skin cells, I mean it's scientifically almost impossible. Yeah.
Really? You've observed that it doesn't go anywhere else? It stays just in those skin cells?
Yeah, unless the skin cells proliferate. I mean, there could be competition, between the engineered cells versus the original cells. I mean, it depends on whether it has a growth advantage or not, but in most cases it will not.
Okay.
Yeah.
So you've observed that it interesting. What about when those skin cells, they die and new skin cells are created? Will they, when they form new cells, do the new cells in that one local area, will they still carry the modification, or will they lose it after a generation or so?
The skin is maintained by we call the basal progenitor cells, and that's also the cells we engineer. And using those the basal cells, we prepare the we call the stratified epidermis for replacement. So in most cases, I mean we cannot rule out that possibility. I mean there will be like migration of normal cells inside the skin graft, or there could be a certain migration of the cells outside of skin graft. But in most cases, for example, from the bone wound treatment, we think it's not very common. Most of the time, the skin kind of follow its we call the upward migration pattern. Is from the basal to the suprabasal layer. That kind of differentiation, more like a kind of a perpendicular way to maintain the tissue architecture. Yeah.
But let's say they don't migrate. There's no migration in or out. How long does a skin cell live for before it is replaced with a new skin cell?
The cells are maintained by the basal progenitor cells. Net cells is kind of a lifelong supply for the to maintain the tissue.
So you said you're modifying the basal progenitor cells. So how often what's the life cycle of a skin cell? It's created by the basal progenitor cell, right? And then it lives for a period of time, and then I would think
the differentiated cells is usually like 1 to 2 weeks. It will slough off. But the basal cells usually stay there for years or even longer. Yeah.
So what happens if when a new cell is created from the basal progenitor cell? Does it still express the same editing?
Yes, if it's coming from engineered edited cells, it will maintain to be the edited cells. Yeah.
Okay, so even when it creates new cells and it divides into new cells, they will still continue to.
They will replicate the genome. Yeah.
Yeah, like when you've done the mouse studies, how long did they have the cocaine resistance for? Did you do it like over a month period or a year? And was there any change in the resistance?
We tested for 4 months.
Okay.
Yeah, and we can still detect a very significant expression of the engineered gene.
Did it change over time? The amount of protein you saw in the blood?
I think it depends on the how well the graft is taken. As long as the graft remain stable, the expression level remain comparable.
Okay.
And that's pretty significant, if I may add, because a rodent, the average lifespan for a rodent is 25 to 26 months of age. So if you can maintain 4 months, and in fact, we have evidence that the skin graft would be stable for up to 7 months or so. Again, depending on how well the grafts were taken. So that's very significant because that's more than a quarter of the mouse lifespan. Translating into humans, then that's 15, 20 some years. That's very significant. So one patch would be good for a very long time.
What about in a situation where you have, let's say, cystic fibrosis? There's a alteration in the coding sequence in one part of the organism's genome, human, and you'd want to affect them systemically in every tissue. You'd want to change that. Could we even begin to do something like that, or is that way far off? Or Can CRISPR not do that?
In every cell, the mutation is corrected. That has to be edited through germline. And in humans, there's a big debate whether we should touch the germline before.
Ethically very controversial. Yeah.
That's right.
Right.
And scientists are working toward it, but until there's strong evidence that CRISPR is absolutely safe before we can touch germline. And there are some illegal research you're probably aware of from certain parts of the world that made news, and that's not a good example.
Right. Yeah, I think say in the somatic cells, I mean, right now will be a good idea. Yeah.
But so there's no way to affect. Let's go back to the skin. So is there no way to alter a patch of skin and have that propagate to all the skin cells, even the same kind of cell? Is there no way to do that you can see?
So actually, there was one paper published in 2017. It's not using CRISPR, but using virus therapy. So that's a skin genetic disease. It's called epidermolysis bullosa. It's almost a lethal skin disease. So it's because the patient has a mutated laminin 3 gene. So the skin epidermis cannot attach well to the underlying dermal tissue. That will cause skin lesion, the blister formation, and also infection because of skin lesion. So in that case, I think that the team collected the skin stem cells from the patient and they used the viral vector to correct the mutation and replace the whole skin using the engineered cells. And that treatment was very successful and saved the patient.
Okay.
So theoretically, it's entirely possible to replace the whole skin if the situation warrants. But in our case, we want minimal skin replacement because why replace the whole skin if a very small piece of skin will do the job, right?
Yeah.
Right. That's what I mean. I just wonder if there would be a benefit if the change propagated throughout all the same cell type.
Right.
It will not propagate to other cell type, but you can technically it is possible to change all the basal to change all the epidermal cells, or we call it keratinocytes. Yeah.
Okay, but so far the viral vector is the only way that people have been able to do that.
No, not necessary. I mean, they use the virus because it's the technology is more mature. You can certainly use CRISPR to do it too.
Why would that work and propagate throughout the other cells when this doesn't? What makes it propagate or not?
No. I mean, the initial procedure is the same. You collect the patient skin stem cells. You do the engineering. You either use a viral vector or you can use CRISPR to modify it. And after you get an engineered pool of stem cells, you can expand those cells to generate enough skin epidermis for transplantation. And you can replace all the unmodified cells in the skin.
So would that mean transplanting the entire skin section by section?
Yes, that's
or
what
could you?
Yeah.
It would be nicer if you could just start it at a certain spot and have it propagate.
Yeah, that will be technically very difficult.
Yeah. Well, that would be the next step, maybe the next gigantic step. If you
Yeah. I mean, yeah.
I wouldn't want to
technical wise, it may be very difficult.
Yeah. Huh. Yeah. Very interesting. So what do you think is going to happen?
In the addiction field, of course, wonderful things will happen. We have tried this approach not only for cocaine abuse. We've tried it in alcohol. We just completed a study with Xiaoyang as part of this. It's fully functional. It worked very well in protecting against or treating alcohol abuse. And we have evidence that it will work for nicotine abuse. And we would like to think that it will work for opiate abuse as well. And also co-abuse of many of the drugs. So in drug addiction alone, the application is potentially going to be very broad, and we hope it's going to produce a very large impact.
Okay, that's true. That's plenty for now. Very good. So what's the best way for people to get in touch and to ask questions and to find out more?
Email both of it or one of us, and we'll try the best we can to respond.
Okay, that's great.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.